All right, guys and girls, we have been working with DNA, and this is your introduction to genetics notes. You'll be using it to complete your study guide for the summative evaluation on Socratic. We will be outlining basically the composition, the structure, the arrangement, the function, and how DNA is basically copied in this section. We're going to make sure that you have more complete notes, and if you have any questions, you will be able to ask me in class during the question and answer section. That's at the beginning of the period. DNA. DNA is the basic genetic material used to compose all characteristics of cells. We know that DNA stands for deoxyribonucleic acid. Its composition. DNA is composed of 3 different chemical components arranged in patterns that make it functional to act as a blueprint of information for all cellular functions. Now, we know that it is composed of a 5-carbon sugar, that is deoxyribose. We know that it has the phosphate molecule that serves as the backbone. Basically, you have your sugar and your peptide or phosphate bond that connect together, and that makes basically the leg of your DNA. And then we have your nucleic acids, which serve as the genetic markers that are used for codons that are used to basically to produce your proteins. Now, the structure of a DNA is, of course, the double helix, as we just said. And a double helix is basically, if you think of a ladder and you were to twist that ladder, then you would have the structure of a double helix. Now, the internal rungs would be your nucleic acids, and they're held together by hydrogen bonds. And then the outside sections, think of the parts between the rungs as your phosphate bonds, and the parts that are connecting the rungs and the legs together, that would be your sugar, basically. You have deoxyribose. It serves as a 5-carbon sugar, and of course the peptide bonds. So we want to know how it is arranged. Now that we know a little bit about the structure, the arrangement of DNA is arranged by corresponding nucleic acids held together by ribose sugar and phosphate peptide bonds. Now we know about that arrangement that when I say corresponding, that we know that A always bonds with T, and C and G always bond together. If for some reason they do not bond together, like if it is replicating, and say a C does not match with a G, it instead matches with a T, that sort of misstep in replication will be known as a mutation, and that's how those occur. The base units of the nucleic acids are guanine, thymine, adenine, and cytosine. It's defined by either by the bond connecting to the 3' and the 5'. Now you need to know the difference between the 3' and the 5', basically because during replication of DNA, it will take place at a certain point, but we'll cover that in the next section of this chapter. Now, and you need to know that it is important for mutations not to take place, and that most of the time they do not, but in rare occasions they do. And it is due to the way that it replicates. It basically breaks apart at the hydrogen bonds and allows for an exact copy between the two parent strands. Now, the function of DNA. DNA's functions as the blueprint of the cell. The majority of the cell's functions take place due to proteins. The building blocks of proteins are amino acids, and different amino acids placed together form different proteins. So basically you have, say you're building a building, you would have a blueprint for that building, and the workers can look at that blueprint and know what goes where, what pieces to put together, how they go together, and things like that. In the same way, in the cell, you have your blueprint, which has all the particular types of proteins that you can produce. So it will go to a certain section of that DNA and it will pull out that particular bit of information and use it to make a certain type of protein. Then that protein will do whatever its function is for the cell. Say if it is a marker protein that tells what the cell might be wanting to bring in, say it needs to bring in some nitrogen or something like that, and it produces more marker proteins, it will know that those will be incorporated in the cell membrane and it will bring in that particular substance. Now, if it is an enzyme that is looking for, say, it needs to break down more fats or something like that, then the enzymes for bile will be produced, and bile will, of course, break down the fats and stuff like that. So basically, you have all the information in your DNA, and then depending on what function needs to take place in whatever cell, it will produce a certain amount of proteins to do that function. How is DNA copied? We said something a little bit earlier about copying from the 3' to 5' end of DNA basically during replication. Well, DNA copying itself is to form 2 new DNA strands, and that is known as replication. It does this by making an exact copy of itself. Basically, it'll rip apart at one end, and it will put corresponding C's to G's, A's to T's, and follow that particular pattern. And corresponding on the other end where the other section was taken apart, sort of like if the ladder was broken apart at the bottom and you started piecing it back together as you went up, you'd form 2 new ladders. This is the process that takes place when the cell goes through either mitosis for cellular division and meiosis when you're talking about reproduction between 2 different organisms, basically. We'll discuss those a little bit more when we get deeper into it. And you will understand a little bit better about how the 2 different processes of reproduction take place, meiosis and meiosis. If a cell reproduces a copy into RNA, which we'll also cover in a little bit later, then it is known as transcription. Like I said, folks, if y'all have any questions about these notes, or if I missed any particular parts that you think might be on the Socratic test, or I haven't made some of it clear, we can cover some of this on the questions and asking section next time in class. All right, y'all have a wonderful evening.